Hello everyone. So today let us learn about gram staining. So gram staining is a widely used differential staining in microbiology, and it can be used to differentiate between gram positive and gram negative microorganisms. So let us move on to the procedure. So in the procedure, first we need to heat fix a smear. Smear is nothing but we will take a lobe of inoculum into the clean glass slide. Clean glass slide means it should not have any kind of oil or grease in that. So we will take a lobe full of inoculum and we will wait for drying. And after that, we will heat fix the smear. And after the heat fixing, we will add crystal violet stain. It is the primary stain in this procedure. And we will wait for 30 seconds. And with this stain, all organism will appear in violet in color. And after this, we will treat it with gramsidine, and gramsidine act as a killing agent as well as the mordant. It means it increases the affection between the crystal violet and the cell. Okay, and it will form a complex. The complex is crystal violet iodine complex. It is known as CVI complex. So after this, all cells appear violet or purple at this stage. And after this, we will treat the smear with decolorizing agent. For the decolorization, we are using 95% of ethanol or ethanol acetone solution. And we will add ethyl alcohol drop by drop until no more color flows from the smear. Excess decolorization will make gram-positive organisms lose stain and give false result. So in this stage, what will happen that the gram-positive cells retained the same color, that is violet color, while gram-negative bacteria lose the CVI complex, that is crystal violet complex. So decolorizing agents act as both lipid solvent and dehydrating agent. We know that gram-negative cell wall is rich in lipid. So what will happen that after adding the decolorizing agent, pores will form and all the CVI complex will escape from the gram-negative cell walls. And after this stage, we will add the safranin. It is a secondary stain. So safranin is a second counter stain. So we will add it after adding the decolorizing agent. So in this stage, we will treat the smear with counter stain of the secondary stain called safranin, and we will wait for 30 seconds. And we know that after decolorization, the gram-negative cell wall does not have any crystal violet. So the gram-negative organism take up this red dye through the pores created by decolorizing agents and appear in red in color. While gram-positive organism, which did not lose CVI complex, will not take up the secondary stain, remain violet in color. So uh, gram staining procedure divides the bacteria into four groups. The first one is gram-positive cells. We know that gram-positive cells will appear in crystal violet in color, while gram-negative do not retain any crystal violet and hence take up the safranin, and we know that it will appear as pink in color. And look at the third group. It is gram non-reactive organism. They do not stain or which stain poorly. Typical bacteria remains colorless to gram staining procedure. For example, Chlamydiaceae, Mycoplasmasiaceae, and Rickettsiaceae. These are the examples of the organisms. And gram variable organism, the fourth group. It means very old cultures of gram-positive bacteria changes in the environment of the organism, slight changes in the staining technique. These are the four groups after the gram staining procedure. So basically, what you need to know that the gram staining procedure is to differentiate between the gram-positive and the gram-negative organism, and the gram-positive cells will appear as violet in color, while the gram negative cells will appear as pink in color. Thank you.